have to say that here is a very nice, so the weather is really nice. So I don't have ice or so we are fine in Seville. Okay, so as Astrid said, I'm going to talk today about the work we have been doing with mayflies during the last 5 years, and that I will continue in my new lab in Barcelona. And so I am, during my career, I've been interested in how diversity, the morphological diversity, is created, and how morphological novelties, and what is the genetic basis of morphological novelties. And for that, sorry, I'm going to move this here. Okay. And for that, so we use the mayfly Cloeon dipterum, and this is the insect. And so why mayflies? So first of all, because mayflies are the sister group to all other winged insects. So they are very interesting in order to understand a very particular morphological novelty, which is the wing of insects. Also, because mayflies are adapted to both habitats, aquatic and terrestrial, as the juveniles live in the water, but the adults are terrestrial and they are flying insects. And finally, and this is what we were talking before, the family of Baetidae has a striking sexual dimorphism. This is the female, as you saw in the previous slide, and you can see the compound eyes, which are located in the lateral part of the head, and the striocelli. So this is a typical visual system of an insect. But when we look at the males, what we observe is that, so they have the compound eyes and they have also the ocelli, but in the most dorsal part of the head, they have this really large eyes, compound eyes called the turbanate eyes, due to this turban shape. So, and those are the reasons why we started working with mayflies. And so my talk is going to go first a bit about how we establish Cloeon dipterum as a laboratory model. And also I'm going to tell you about the genome project and what we discover sequencing the genome of Cloeon dipterum. So as I said a bit before, so the first challenge we encountered when we started the project is to close the cycle. So we wanted to have an inbred line in the lab, and the first problem we encountered was to close the life cycle, as mayflies mate in swarms, and then so the female lays the eggs in the water, the eggs hatch, and the nymphs molt several times in the water until they reach a particular size that they molt once, but these times they leave the water and they molt as a sexually mature subimago that molts once more into an adult or sexually mature adult, and then they mate and the cycle is closed. And the way they do that is forming swarms of thousands of individuals. So this is a very nice picture that won a Royal Society prize several years ago. And this is a video that I took from the internet that is showing a weather satellite showing a swarm of mayflies forming in the Mississippi River in the States. So you can imagine that for us it was a bit difficult to close the cycle, because we didn't have, of course, a space to leave mayflies flying freely in our institute. So we had to use a bit our imagination, and with this drawing that a collaborator showed us, a paper from the 70s, where they have this drawing, which so is easy in the drawing, but so it was a bit more difficult in reality, because they don't like at all what is called forced copulas. But at the end we managed, and here you can see, so this is this is a couple mating. So what we do is just to put a male and a female together, and with a bit of or a lot of struggling, finally, so we have these matings. And one of the characteristics of this species, Cloeon dipterum, is that they are oviparous. So when we manage to get these 4 scopulas, what we do is to keep the female, and we keep the female for 15 days, which is the time that the embryonic development lasts. And then after 15 days, we put the female on the surface of water, and then she starts laying the eggs. And you can see here how she's laying the eggs. And as I said, that they are ovoviviparous, just after laying the eggs, they immediately start hatching and swimming. And here you can see a higher magnification, and you can see more clearly how the eggs hatch, and you can see already all the eyes and the ocelli, the compound eyes and the ocelli already formed. So that was nice, because so we managed to close the cycle, and this allow us to have easy access to many individuals, as many as we want, because in every clutch we have like from 100 to 500 individuals that are synchronized. So it's nice, so we can just perform embryonic studies, or we can inject some dyes, and so we can study, and this is one of our projects, to study the origin of the turbanate eye in males, and also we can investigate which are the genomic signatures, or how the genome and the transcriptome of mayflies adapted to these 2 different lifestyles. So now I'm going to go into the genome project, and for that, what we did, so we sequenced the genome of a male, and we use 2 different strategies. We use Illumina and also nanopore sequencing, and I'm not going to go much into the details, but if you want, you can ask me later on. And we also sequence with Illumina a female, just to see the sex determination components of the in the genome. Sorry. So we didn't only sequence the genomic DNA, but also we profile multiple samples of RNA samples of different developmental stages, embryonic stages, or different tissues in nymphal or adult stages. So muscle, brain, gills, ovaries, etc., and also several stages of heads during the nymphal stages. And why we did that? So first, to help with the gene annotation of the genome, but also because we were interested in investigating a particular or the gene expression across these different samples. And here, so you can have one example. And this is abdominal A, which is one of the Hox posterior genes. And this is how it looks in the browser. And so the Hox genes, as you may know, so they are expressed, the anterior genes are expressed early in development, and the posterior genes are expressed a bit later in development. And so this is a posterior Hox gene. So when we look at the transcriptomes, what we observe, as we expected, is that in early embryos, the expression of this Hox gene is low, but as embryogenesis progresses, it does also, the expression of this gene increased. But of course, so this is just one case, and we wanted to do it in a more genome-wide way. And for that, we use this other type of analysis, which is called M-Fuzz soft clustering. And this clustering, which allows us, is to profile genes that are expressed along our different samples. So for that, we use the 4 embryonic samples that we have, 3 nymphal stages, and also the adult. And this, so for example, this is just an example of one of the clusters we found. And for example, this cluster shows or has genes that are highly expressed during the embryonic stages, but they are not expressed so much either during the nymphal or the adult stages. And so this is an example, but so we can recapitulate so different profiles and different types of gene expression. And at this point, what we wanted to focus in genes that could explain or that could recapitulate these modules that the different the life history traits of mayflies. So genes that, so these clusters that have high expression during embryogenesis, other clusters that contain genes that were highly expressed during nymphal stages, or clusters with genes that are highly expressed in adults. And so we have this, and we focus a bit more in the transition between aquatic and aerial or terrestrial life. And when we looked at so we did a gene ontology enrichment analysis, and when we looked at the terms that were enriched in each of these clusters, what we observed is that when we look at the genes that were highly expressed during nymphal stages, we found enrichment of terms related to perception of chemical stimuli or odor unbinding. Whereas when we look at the terms that were enriched in clusters that contain genes highly expressed during adulthood, what we observed was an enrichment in terms related to vision. So something that we say in the lab is that so the nymphs smell the environment, whereas the adults see what happens. So we wanted to focus a bit more then into this gene ontology enrichment and what happened with the chemoperception. And for that, so one thing that we had to take into account is that depending on where you are, so if or where the nymphs are, if they are in water or if they are in land, so they have to have physiological, anatomical, and molecular adaptations in their chemosensory system. So it's not the same. So the way we perceive the smells or the chemicals is completely different if we are on the land than if we are in the water. And another important thing that we had to take into account is that if we look at the different gene families or chemosensory gene families that are in insects, what it is known is that, for example, one of the families, the odorant receptor gene family, appeared for the first time in Insecta, and the odorant binding protein gene family appeared for the first time in Hexapoda. So those are innovation of these two lineages, and as this, so it has been suggested that these two gene families appear as an adaptation to terrestrial environments. So crustaceans don't have it, so don't have them, so they live in the water, and when insects conquer the land, they also, so as an adaptation, they evolve these two new gene families. So with this information, we started to think what happens with these gene families in our mayfly. And so we and our collaborators study so which is the complement of each of those families in mayflies, in Cloeon dipterum, and what we observed, and it was quite striking, is that for the odorant binding proteins, Cloeon dipterum has a huge expansion. So we found 171 genes of odorant binding proteins, whereas, for example, if we compare with Blatella germanica, which is known to have many expansions of all these different chemoperception gene families, it is so Blatella only has 109. So it has even more genes than Blatella. So it's a really, really large expansion of this gene family. So the next question we wanted to ask our genome and our transcriptome is where these genes are expressed. And for that, we went back to our M-Fuzz subclustering analysis, and we looked in which clusters the chemosensory genes were located. And what we found is that most of the chemosensory genes were located or placed in these 4 clusters. And when we looked which kind of clusters they are, what we found is that 3 of them are clusters that contain genes that are expressed during nymphal aquatic stages, and this cluster number 10 that is not an aquatic cluster, but it has genes that are highly expressed just at the end of embryogenesis. So these embryos are about to hatch into the water. So this is already telling us about that at least these genes are expressed during, or at least the mayfly could use them to live in the water. So not only that, as we have many different samples, we also look across these different samples where these odorant binding proteins were expressed, and we found that most of them were expressed in these samples that were nymphal heads, it was quite expected, because insects used to smell or have the chemosensation through the antenna in the head, so that was quite expected. But what wasn't so expected was this cluster of odorant binding proteins that were expressed in this other tissue, which were the gills. So the gills in mayflies are these abdominal structures that are located, and they located and they pump them. And so this movement and what has been described until our study was that the gill plates in the nymphs of mayflies were used to breathe. So that they were a respiratory organ. But it looks like it is not only a respiratory organ, and probably it also has a sensory function. And indeed, when we look at the expression of some of these odorant binding proteins, we observe that they were expressed across the entire gill plate, and that this expression colocalized with HRP, which is a neural marker, suggesting that these gill plates or these abdominal gills are not only a respiratory organ, but also a sensory organ. So this is what we found out about the chemoperception during the aquatic stages, but what happens with the visual perception of adults. So for that, the most essential thing to look for is the opsins, which are the light-sensing molecules. And in insects, there are 3 main families: the long-wave sensitive opsin, the blue sensitive opsin, and the ultraviolet sensitive opsin. And these opsins have undergone different expansions or reductions independently in different lineages. So for example, the long-wave sensitive opsins duplicated several times independently in different lineages. So what happens when we look at a Cloeon dipterum genome? So what we observe is that it also has an expansion of the long-wave sensitive opsins. But what was more interesting was that it has also an expansion of the blue sensitive opsins and the ultraviolet sensitive opsins. And with 4 different opsins, it was a really, really large expansion that hasn't been described so far in other organisms. So when we looked a bit more into the evolutionary scenario of these expansions using other data available, what we observed was that the long-wave sensitive opsins duplications were ancestral to all the Ephemeroptera lineage, But when we look at the blue and the ultraviolet expansions, what we found is that they were ancestral to the Baetidae family. And the Baetidae family, if you remember from my first slides, has a very striking trait, which is the turbanate eye. So at that point, we wondered if the appearance of this new visual system had something to do with the appearance of these new opsins. So one easy way to test that, and because we had transcriptomic data from males and females, what the first thing we did was to check the differentially genes, differentially expressed genes between males and females, as females don't have turbanate eyes. And not surprisingly, at this point, what we found is that the two genes that are more highly expressed in males, and actually in females are not expressed at all, were one of the blue opsins and one of the ultraviolet opsins. So we check using, so at this point we knew that, or we learned that they were male specific, but were they turbanate eye specific? So we did some in situ hybridizations, and here you can see, for example, ultraviolet, the UV opsin 2, which is common, so it is equally expressed in males and females, and we observe that the female express it in the compound eye normally. When we go to this male-specific opsin, the ultraviolet opsin 4, the female doesn't express it as we expected from our RNA-seq data. When we look at the equally expressed UV opsin 2, it was expressed in the compound eye, the same as the females. And when we look at the male-specific opsin, the opsin 4, we observed that it was only expressed in the turbanate eye and not in the compound eye. So indeed, this opsin at least is male-specific. Sorry. And finally, so as I said at the beginning, mayflies are really interesting to understand the origin of wings, as they are the sister group to all other winged insects. And we wanted to have a first approximation in a transcriptome, so using our transcriptomic data to this question. And for that, we use a different type of analysis, WGCNA, which is Wave Gene Correlation Network Analysis, which is another type of clustering. This time, what we have is all our transcriptomic data from the different tissues, and what we get here are clusters of genes that are co-expressed in these tissues. And we did that for mayflies, and we also did it for Drosophila and Strigamia maritima, but I'm not going to talk about this Strigamia data. But so we did for this, so we have several clusters or modules in this case of genes that are expressed in particular tissues for mayflies and Drosophila. And what we did that was to do a pairwise comparison and to check which kind of genes were located in these clusters and if they were the same. So for example, and we have for many different type of clusters. So we have for muscle, for ovaries, brain, gut, etc. But of course, we were interested in first if we had a wing module in mayflies and flies, and if it was an overlapping of the genes. And so here, so we have a cluster that was the wing cluster in mayflies and also in Drosophila melanogaster. And when we look at the pairwise comparison, we observe that they highly correlate. So actually in total, we have 126 ortholog genes that are shared in these two wing modules. And when we looked a bit in the type of genes that were located in these modules, we saw that they were genes that had already a described function in wind development, like Wind 6, Hertz, Fujitaratsu, sorry, for jointed, Yorkie, etc. But also we found a bunch of genes that had an unknown function. And when we took from these 40 genes, we took a subset. Actually, we checked 8 different genes, and we, using Drosophila this time, we did knockdown experiments using RNAi specifically in the wing, and we observed, we checked if we had a phenotype, and actually we did. For the 8 different genes that we tested, we found a wing phenotype. So it's not so. Here we can see that there is not only a conservation in the expression, so it's not only a transcriptomic conservation, but also the function is conserved between Drosophila and Mayfly wing genes. So, and again, this is just for the conservation of the transcriptomes and how similar are nowadays the transcriptomic programs of wings in these 2 lineages. But what is the first origin? So I'm not going to go much into the details, but now there are several hypotheses suggesting which was the structure that gave rise to wings. And so the most accepted theory these days is a dual hypothesis in which parts or regions from the pleural region of the insect fused with some regions of the most dorsal or tergal region of the insect, in the thorax. So one of the hypotheses is that one of these pleural plates or structures could be the gill plates of mayflies. So with this in mind, we wanted to know how similar the transcriptomes of the gill plates were to the transcriptomes of wings. And for that, we did first a clustering, and in fact, so from all our samples, we observed that our wings and gill transcriptomes clustered together, which was already a good hint. But another analysis that we did was to check genes that were specifically expressed in wings and a second tissue, and see how many times this second tissue was the gills. For example, this is one example. So we have here the one gene in, which is highly expressed in wings and in a second tissue, which it happens to be gills. And we see that in the rest of the transcriptomes, it is not highly expressed. So how many times we have this? So in total, we have more than 40 genes, which is actually more than 40% of the genes that are highly expressed in wings are also highly expressed in gills, suggesting some similarity between these 2 transcriptomes. And so we cannot say yet that they share a common origin, but at least at this point, and as a first approximation, we can say that so they have a similar genetic program and a shared transcriptomic program. And with that, I'm going to finish just to summarizing a bit what I showed, that Cloeon is I hope you are convinced now that Cloeon is a new model to study evolutionary novelties that contrary to current views, odorant binding proteins, so this chemosensory gene family, play a main role in aquatic contexts, that gills function as sensory organs, that the UV sensitive Opsin 4 is turbanate eye specific, that there are conserved sets of genes between Drosophila and Cloeon, that some of these conserved sets of genes are wing genes, and that when we knock down them, they have wing phenotypes, and that gills and wings share a transcriptional program. And finally, I want to thank Fernando Casares and the lab, who helped a lot at the beginning, especially the people here that were students working with mayflies. Also, as you can imagine, the genome project was a big collaborative effort, so there were many people involved in that, and also Dave Funk and other people from the CIBD that were helping at the beginning setting up the culture and the funding bodies and for your attention. And as Astrid said, I will be starting the lab soon. So if you are interested in working with mayflies, so you can drop me an email and we can discuss funding opportunities. Thank you.